reducing morbidity to therapies is validated, and that through real life or real world experiences, this can then lead to, over time, improved therapy. And when we say over time, we're not talking about 1 year, we're talking about a decade, most likely. So I think that's an important point. The types of data that we'll hear about today are not just genomic data. There's all sorts of data. And I'm going to emphasize in the beginning the importance of research data, which I think is very important to making the observations for changing clinical care. So this has been obviously something that I've thought about for a long time. I just went back into archives to find this commentary that I made, where the main point is that for us to make advances in precision medicine, we really have to combine the clinical data with the genomic data and make this widely available. And so I think that's an important point. As pathologists, how do we do this? And really with the focus more, let's say, on the genomic tumor data, cancer genomic data that we use, how do we do that? So a few of the important points that are interrelated to the bioinformatics. So obviously bioinformatics is going to play an important role, but it's going to require having high quality samples that are available. So this could be patient samples, or it could be archival samples, developing molecular signatures using very variable genomics approaches that we'll hear about, and then having these validation cohorts that are available to make observations. And then finally, making the data available, not just for clinicians for clinical care, but also for researchers doing both basic science, but also translational work, and maybe even clinicians thinking about designing clinical trials and what sort of correlative science. And I just gave examples of a few ways that data can be visualized, particularly Oncomine, transcriptomic data, and CBioPortal, which are used heavily in the research and also in the clinical communities. So just a few comments now about, now I'll focus on prostate, but this is probably going to be true for other diseases as well, which is that we have to recognize that at the germline level, there's a tremendous amount of human diversity. And a number of years ago, we started looking in the Tyrol population where they'd done PSA screening. And just as an initial comment, it was very important to look at the population, as mostly European, and look at different polymorphisms. And this study looked at copy number variation, but polymorphisms in a population are going to be important and will help understand response. These may be subtle differences, but it's important to remember that when we're thinking about the bioinformatics, we also have to think about the germline alterations that may play an important role. Our work for a number of years, working closely with the Roel Schneyen's group, has been focused on the genomics of prostate cancer. And I think in the very early studies, this was using spotted arrays, so the real ancient history now. But basically, the idea was to look for transcripts that are overexpressed in prostate cancer that are not overexpressed in benign tissue. And this led to a number of observations, and I would just highlight ERG, which I'll talk about in a moment, which is the ETS transcription factor that came up as one of the interesting genes. This type of work, and then a number of studies that followed, represented an opportunity. So we saw there were a number of transcriptomic data sets. And so first bioinformatic opportunity here was say, let's combine the data and think about a meta-analysis map. And so this study, published in 2002, was a precursor of what became Oncomine database, which was a database that looks at many different cancer types and allows you to ask the question, what genes come up and consistently, regardless of platform, in a number of studies. And one of the top hits that we had for prostate was something called AMACR. And this is a gene that in prostate, when you look at prostate tissue, is highly expressed in the tumor, but not in the benign. It's also expressed in other cancer types, but for prostate, it's very specific. And this was an opportunity to identify something that's now used clinically to identify something that is cancer versus non-cancer in the setting of prostate. this led then to also a number of studies that I'll highlight just a few aspects of, where we wanted to think about prostate cancer as a collection of homogeneous subtypes that are identifiable by various molecular criteria. And the starting point would be thinking about leukemias or lymphomas, where you have many, many subtypes that are really defined molecularly. And these have come out through various types of genomic and transcriptomic and other types of omic studies that have helped supplement what we know clinically. The problem, of course, with this is that similar to these classifications of leukemia or breast cancer, it's very often the fact that if you don't have a specific treatment for that subclass, it's not of great interest, often clinically. And our view was, what we argued was that having classification was required for interpretation of how patients respond. And then we could then potentially develop targeted therapies. And I just would highlight this as an important concept, because it sort of suggests that genomics and other types of analyses may precede the treatment, and that these may also guide the treatment. So it's not always the case that it's the reverse, where you have a treatment and then you're trying to figure out who should be treated. You actually may need to find the subclasses to then help define new therapies. So this led to a series of studies in order to try to classify prostate cancer. And I think one that now also is relatively simple from the perspective of bioinformatics was that we took a number of prostate tumors that we had, where we had transcriptomic data, which we had already published. So this was existing retrospective data, and applied a transformation where we were looking for oncogenic outliers. And this was called COPA. There are a number of ways to do this, very simple approach to really looking for outlier profiles. And when we did this, what came out in a number of the studies were genes that were related to gene fusions, as well as that's transcription factors in prostate cancer. And so this led to observations, for example, the fact that these overexpressed ETS transcription factors were mutually exclusive, and that when doing RT-PCR experiments where you look for potential fusion partners, that TMPRSS2, which is located in chromosome 21, could be found to be associated with any one of the S transcription factors that we found to be overexpressed, suggesting that we had found a fusion of an androgen-activated 5' partner and a downstream growth factor, which is S transcription factor. And then in further studies, we were able to use SNP arrays. So this is, again, very old-fashioned technology at this point, probably, and demonstrate that there was a 3 megabase deletion that was leading to this gene fusion in prostate cancer. So fast forwarding from these original observations made through very simple bioinformatics examination on relatively simple samples, I think this is an old slide, but I mean, many, many thousands of cases have been evaluated both for research and clinical, and approximately 50% of prostate cancers have this gene fusion. So it makes it a very common fusion. Currently, there's no therapy, but we do have very good immunohistochemistry assay that we developed to identify cancer and neoplasia versus benign. And so when the fusion occurs, this is a cancer-specific, prostate cancer-specific event. This has also led to the opportunity to develop commercially assays to identify cancers. Now, I think the important point I wanted to bring up here is that this is a very nice marker for a subclass of prostate cancer, but there's no current treatment that's associated with that. This initial type of observation led to the thought that we really need to systematically look for gene fusions in cancers. And with the advent of RNA sequencing that had just started, so basically next generation sequencing, we and many others were able to develop pipelines. Just an example of one pipeline, FusionSeq, to identify new gene fusions. And this type of work led to the identification of rare fusions that were potentially targeted. And here are just two examples of BRAF targetable gene fusions in prostate cancer. And this led us to develop a map of really all the fusions we could identify. Almost over 50% of prostate cancers could be defined by some gene fusion. This approach also led to identification of other gene fusions in other cancer types, and sometimes defining a specific fusion that would define a specific subclass of cancer. So these are the types of informatics that are really required, I think, to make the discoveries, even if they precede any clinical application. Now I'd like to turn to another aspect of precision medicine I think is very important and highlighted by Harold Varmus, who pointed out that in this US study, but I think it's generally true, which is that providing support for genomic sequencing, could be transcriptomic, could be proteomic evaluation of cancer is extremely important, even prior to the obvious clinical application. The reason being is that we need this information to inform us how to better care for patients. So again, the concept that we have observations that we make prior to understanding how it's going to affect therapy. And I would just give one example from our work. Again, I'm focusing on prostate and things that we've done so that we have a lot of knowledge about, but obviously there are many other examples that could be given. This is an example where we really start from an extremely small number of cases, so a whole genome sequencing of 7 localized prostate cancers led to the observation that 2 of 7 harbored a recurrent mutation, or what we thought was a recurrent mutation of SPOP mutations. I won't go into the details of the gene, but the point is that when we then extended it to larger data sets here in 112, we found approximately 13% of the cases had that. And then a few years later, the TCGA conducted a study where they demonstrated that it was about 11%. So we're reaching some stability as to the frequency of this mutation. It's a mutually exclusive from the ETS fusion. So obviously you have to get to many hundreds of cases to get to the point where you can say that something is mutually exclusive if it only occurs at around 11%. Now, there's no treatment that's associated with SPOP subclasses, but I just want to highlight that fast forwarding a few years, there have been 2 clinical studies now that demonstrate that the biology of the SPOP mutant prostate cancers is different than the ETS or the non-ETS tumors, in that they're more sensitive to various therapies. And this is the hormonal therapy. So I won't go into detail about that. So the important point is that by classifying cases, even prior to known therapies, it can be very helpful. The last point I'd like to make, at least for purposes of the precision medicine aspect of this talk, is just that reporting of complex data is going to be one of the highest, I think, goals that we're going to have. And bioinformatics obviously plays a very important role. So this is dating back to 2016, and prior we were at Cornell, we were doing whole exome sequencing, so very extensive sequencing of patients with advanced cancers, not just prostate cancer. And we had to then develop pipelines for this, as I'm sure many of you have now developed sophisticated pipelines for analyzing advanced cancers and other cancers, and then developing a way to report this. So I'm not showing this as the ultimate way to report, but just the point is that we have a very simple report that can go to a patient with extensive genomic data behind it. But then we also had to think about how to report and to maintain the other data. And in our instance at Cornell, we tried to maintain all of the data, even if it was, let's say, research grade, where there was no known clinical implication. So the point being is that the reporting of the data to both clinicians, but also maintaining it for translational research is extremely, extremely important. So what's going on in Switzerland, and just very briefly with the SPHN program, we have a focus to try to also take genomic data from a number of centers, and a number of you on this call or attending the symposium are involved in this. And this is the concept that there are various platforms, different universities, university hospitals, but also non-university hospitals in Switzerland, where either Illumina panels or whole exome sequencing, RNA sequencing need to be evaluated in a systematic way. And so the goal of our project, at least for genomics, which is called SOSIP, is to uniformly analyze the data. We would like to then be able to present data in the consortium, but then also eventually publicly in a platform that's easy for researchers to evaluate. So it could be a number of platforms. Here we're showing CBio Portal. I know there are other platforms developed at Zürich and other sites, but this is an easy way for a visualization of the data. And then one other, I think, important feature is that by doing this, and I think this is an important component for what we're doing in bioinformatics and context of precision medicine, is it also allows us to then participate both nationally but also internationally. And this is a image of our proposed international ICGC Argo project from Switzerland that's co-led with Gunnar Retsch, who's one of the lead computational people in the SOSIP project, as well as Charlotte Yang, who's at Bern. So that's the end of the first part. I just wanted to give some real background about where I think bioinformatics plays an important role. So I'm obviously a very big supporter, but I did vote in the beginning as a neither. So I'm not a bioinformatician and I'm not per se a clinician. I'm somewhere in between. So our lab has actually been very much focused in basic and then translational aspects of prostate cancer. And the next part, the second part that I'd like to discuss, and I'll discuss it very briefly, but I just wanted to go over the concept of lineage plasticity. And I think this is extremely important because it's something that's coming out of precision medicine. And the general framework is that we're identifying patients with cancer. There are targeted therapies, and that most often when you target a specific pathway, either the index effector or the alternate effector oncogenic output is altered initially, but then resistance occurs. And most often the mutations or alterations that allow the pathways, these oncogenic output pathways, to reactivate, the mutations occur here. But what we're also observing now is that there are alternative or pathways that are basically indifferent to the main states. And this is leading to the development of a lineage plasticity, or really the changing of what the tumors look like so that they're no longer dependent on the initial output. In prostate cancer, as a specific example, androgen receptor signaling is critical for the cell growth of tumors, and that's the mainstay for therapy. So most of the therapy is focused on targeting AR, and most of the alterations that we find are mutations, amplification, splice variation that occurs. What's come about more recently through precision oncology trials is the observation that we see lineage plasticity and the emergence of what we refer to as neuroendocrine prostate cancer. And I'll just highlight this as an example. So here's an adenocarcinoma. Patient was treated with anti-androgen therapy, and then 2 or 3 years later comes back now with this blue tumor, which is a small cell cancer. So something has happened. It could be due to heterogeneity, it could be due to sampling, but what we're seeing pretty consistently is that there is a transformation in a subset of cases. And why this is important is because with androgen deprivation therapy, patients can be maintained for quite a while, but once you develop the neuroendocrine cancers, there's no standard of care therapy. So this is really represents a precision oncology opportunity to try to identify new therapies, new ways of diagnosing and treating patients with advanced disease. So what do we know about this? I think taking a very similar approach that I described earlier, we wanted to ask questions a number of years ago just to understand the basics of what these neuroendocrine tumors are. And we used a multi-omic approach. Again, this is circa 2010. So a lot of the technologies are older technologies. I mean, I think single cell sequencing would have been part of this at the time, but we looked at methylation, RNA sequencing, and whole exome sequencing of these cases to try to ask the question, how is this lineage plasticity occurring? And there are different possibilities. So the first question is, do we see any obvious genomic differences in the classes? And what we show here, we superimpose the adenocarcinomas with the small cell, we see that mostly they overlap, and there are a few areas of differences. So P53 and RB loss tends to be more frequent in the small cell, but otherwise there's no major differences. We use the transcriptomic data to create a signature. So here's where bioinformatics was very important. We asked the question, can we, using this training set, develop a signature for neuroendocrine signaling and also androgen receptor signaling? And then we would query a number of available data sets. This is an example where having a genomic transcriptomic data sets publicly available and available to your consortium are important. So as expected, many of the localized tumor data sets had very low frequencies of these neuroendocrine tumors. And in one of the advanced cohorts from Cornell, we could see around 10%. So we were predicting that around 10% of these treated tumors have this neuroendocrine phenotype. Then fast forwarding to a Stand Up to Cancer, Prostate Cancer Foundation funded clinical trial where here we show the results of 400 and approximately 50 cases where we look consecutively at patients treated with advanced cancer in a clinical trial where we did whole exome sequencing and RNA sequencing for metastatic sites of all the patients. We found that approximately 10% of those patients developed small cell or neuroendocrine prostate cancer. These are patients who are initially diagnosed with adenocarcinoma. So we suspect that there was a transdifferentiation or some change in their tumor type. And then using the scores that we developed, we then said, well, what do the tumors look like when we compare the clinical assessment of the tumor to the genomics? And I just want to point out what we might expect to see. So in a quadrant where we have very high neuroendocrine scores determined by the neuroendocrine score that we developed from the transcriptomic data, we might expect to see a small cell. And in the area where we see high androgen signaling but low neuroendocrine, we would expect to see this. And potentially there would be other types of cancers we would identify. And this is what happens when we superimpose the real world pathology evaluations. So in the light color is the adenocarcinoma, in the darker color, we have the small cell. Again, this is the assessment by pathologists who knew nothing about the genomic data. And for the most part, they appear to fit into the quadrants, but not perfectly. And I would just highlight that here's an example of a tumor that would not be called necessarily small cell. It's an unusual tumor, but it wouldn't be called a small cell tumor, and it fits into that quadrant of small cell. And here's a case that would be very highly likely to be called a small cell, and it's not. It fits into this adeno phenotype. So clearly, we have information that tells us that most of the cases fit nicely into one quadrant or the other, but we're missing information. And why that's important is because the patients who are being treated will be treated differently if they're put into a category of neuroendocrine prostate cancer. If they have neuroendocrine prostate cancer, it may be that some oncologists would not continue to treat them with androgen receptor signaling inhibitors. And what I think the study shows is that although that may be not a very effective way of treating the patients, it still may be effective. And as we saw, there are some cases here in the quadrant that are very sensitive to AR inhibitors. And so I think one of the first lessons from this precision medicine project is that we have to really rethink how we categorize just using clinical approaches and even some relatively sophisticated genomic and transcriptomic approaches. The continuation of the study was to then say, well, what are some of the inputs of, what are some of the important aspects of some of the other components? So epigenetic transcriptomic and genomic components were evaluated in a sort of principal component analysis, and methylation was demonstrated to be very important. And so the only thing I would highlight here is that EZH2, so a gene involved in the polycomb complex 2, is highly expressed in the neuroendocrine tumors. And this suggested to a number of researchers to potentially target this epigenetic alteration. So there are inhibitors for EZH2. And this led to several studies, which I won't go into detail, but just to say that there are clinical trials that are based on observations that EZH2 is highly expressed and may be a way to modulate these cancers. The other research-based observations are with the observation that P53, RB are altered, can you model this system? And so Owen Whitty's group did a forward modeling approach and was very nicely able to show by using RB, P53, but also a loss of PTEN or overexpression of AKT, they were able to show that they could model the system quite nicely and recapitulate neuroendocrine tumors. And this is what's shown here. I think the important point is that then from a model system, then you can return to the genomic data, transcriptomic data, and show that the model tumors either in transcriptomic space or using a TAC-seq in epigenetic space actually fit in very nicely with the human tumors that have been characterized. So I think this is an important role of bioinformatics, is that even if it's going from a model system, we want to use clinical data sets to really have a reality testing to see whether these are in any way related to human diseases that we see. The observations I made about prostate are also true about other cancers, and this is just an example of lung cancer where it's known to be true that a lot of the same changes that I spoke about are true for lung cancer. So in the last few minutes, I just want to discuss very briefly a specific epigenetic observation that we made related to a SWI/SNF complex. And I'll just highlight here some of the bioinformatics aspects of this. And the idea is that we asked the question whether one of the components or the entire complex of SWI/SNF that regulates very much the opening and closing and the movement of histones. So in contrast to the polycomb complex, it helps open up regions of the genome for transcriptomics. And whether we could define a neural complex that might explain why we see some of the changes we see in the context of therapy. I won't go into all the details here. This is published work, but I just wanted to highlight that being able to use publicly available data sets, and these are just the numbers here, we're able to first confirm that we don't see any recurrent mutations. We did see transcriptomic changes in some of the polycomb genes and some of the SWI/SNF genes, and in particularly in the neural differentiation genes. So BAF53B here, for example, as well as SMARCA4. And then we could confirm this with available tissue samples. So again, validation is very important. And I think that's why the close interaction between the clinical and translational or basic observation is very important. And here you can see just for the BAF53, it's very highly expressed in their endocrine tumors, but not in the other types of disease that we looked at. One other aspect, which I won't really go into too much today, but obviously recognize that we need to be doing single cell, is that if you look at heterogeneity, I mentioned that SMARCA4 or BERG1 is very highly expressed in these tumors, but also if you look at its paralog, SMARCA2, you can see that it's also expressed in a mutually exclusive manner, which can be seen under the microscope, but also if you're doing single cell sequencing, we would expect to find two different populations. And so that would be something that probably would be extremely informative. We can model this in various cell lines, and these are just some cell lines that are neuroendocrine. You can see they have very high expression in the BAF53. And then we can knock down these cell lines for the various components. And I'll just show you an example where we knock down SMARCA4. And from that, you can then ask general questions. And these are things that commonly are done in collaboration with bioinformatics support, where we ask questions about what are the alterations, we can then develop a signature. And so this was done in the recently published study where we said, let's have a signature for what happens if you knock down SMARCA4. And in fact, when we do that, we found in applying this to clinical samples, you can find that it's highly, highly enriched in these neuroendocrine tumors. So going from observations in cell lines, knocking down a gene, so we know it's only that gene, we can see that there's some reality testing, because it looks like the tumors that were evaluated in around 140 tumors here show the neuroendocrine features. And when we then extend this to even larger cohorts, and this is over 5,000 cases that were available to us to query, we were able to find that this SMARCA4 signature, high SMARCA4 equivalent, is associated with a higher risk of disease progression. So I think this is the main point I want to make is that going from observations, it's very important to try to find the cohorts and have the cohorts available for validation. There are also opportunities, because there are a number of drugs that are available, and this is just an example of unpublished data where we've taken a protein degrader for the SMARCA2 and SMARCA4, so for the ATPases of this SWI/SNF complex, and can demonstrate that in some model systems, the model systems are sensitive to the degrader, but in other systems not. So having model systems is also going to be very informative. And I think there may be discussions today about organoids or other approaches, but clearly having model systems available to test drugs is going to be very important. Finally, using proteomics, one can also ask the question, what are the genes that are associated with this complex? And this study, one of the things, in addition to being able to pull down other members of the SWI/SNF complex with the goal of trying to define the specific neural complex, we're also able to identify a number of interesting genes, including Yamanaka factors, so SOX2 and KLF4, which are known to be associated with pluripotent stem cells, suggesting that this subtype of prostate cancer that's undergoing lineage plasticity may be a stem-like phenotype. And in just very early observations, we were able to see in the organoids that we grew, there's a heterogeneity. And this, just to focus on the SMARCA4, we see SMARCA4 expression in the context also of tumors that are expressing one of the Yamanaka factors, SOX2. And then you can see here that, in fact, you can see there's a population of SMARCA4 overexpressing neuroendocrine cells that also are expressing SOX2. And then now in unpublished studies from our group, we're able to demonstrate using two approaches, but just to highlight here, a proximal ligation assay, where you can see both expressed in the same cells. So I think it's important that going back into the lab and demonstrating the reality of what you find is very important when you're using these high throughput approaches for discovery. And just to end, recently there was a paper published by another group independent of us where they use inhibitors of the SWI/SNF complex, and they demonstrate, I think very importantly, that one of the types of genes, so the Yamanaka factors, OCT4 and SOX2, are really immediately altered as far as the occupation sites when you look at ChIP-seq or other types of studies looking at chromatin availability when you knock down the SWI/SNF complex. So this is very suggestive that we're looking at a stem-like state. So this is the conclusions. I won't go over that right now, but just to say that now we're thinking in terms of lineage plasticity occurring in the context of genomic changes, but not only that, also epigenetic and probably also microenvironment changes, which I didn't discuss today. And obviously, we also have to think in terms of the context of heterogeneity. So there's just an image showing the same patient's tumor where you can go from adenocarcinoma to neuroendocrine. So just remembering that all that we're seeing is also in the context of a tumor that may be very heterogeneous. And finally, that we may be seeing in the course of therapy a stem-like state that can be driven maybe to an end differentiation of neuroendocrine, or it could also be other types of differentiated states like sarcomatous. But this potentially opens up an opportunity for therapy, because could we reverse this back to an adeno or luminal state? These might be potentially targetable. So with that, I'd like to conclude. Just want to highlight some of the people who did a lot of the work on the SWI/SNF work, which is Joanna Sierda, Philip Thierga, and also Anke Ausbach, who are in Bern. And then I also highlighted a lot of the computational work that was done from early work with Francesca Di Michaelis, as well as Levi Garraway's group, which is not listed here, for a long time collaborator, and more recently with Salvatore Piscoglio from Bern. So I'd like to thank you for your attention, and happy to take questions now or during the panel session if it's time is available. I'll stop sharing.
Brilliant. Thank you for that talk, Professor Rubin. So if anyone has any questions, put them in the Q&A section. We actually have one so far from Joachim, if I'm saying that right. Is there ongoing work to transform any of the genetic knowledge that you have showed in your presentation to cancer therapies?
Yeah, so that's a great question. And there are a number of avenues. So mostly we're not a drug discovery lab. So what we've done is we've partnered with large pharma and biotech, and we have taken some of the observations. So some of these are quite far along. So I'll just give, I won't discuss the company, but I'll just give an example. So with the neuroendocrine tumors, because they're so different, we have been looking at cell surface changes, what proteins are expressed specifically on these transformed tumor cells. And that's the basis for an immunotherapy project that is quite far along. We've been working for about 5 years on that project. We have other more nascent projects that we're doing actually in the academic setting, University of Manchester, but also with other commercial companies related to SWI/SNF modulation, and that is looking at some of the components to modulate them. So I think great question. I think I'm sure we have people who are much more expert in this, but we're in an area that's not lung cancer. So there are not a lot of recurrent targetable mutations. This is much more difficult. We are dealing with transcription factors and others. So it's been a challenge in prostate cancer when you move beyond P10 targeting and androgen receptor targeting.
Okay, great. So I know that the SWI/SNF complex is also kind of implicated in renal cancers as well, with PBR1 being like the largest. I don't know, are you looking into renal cancers, or is this focusing on prostate?
Well, this specific, so I mean, around 20% of all cancers have some mutation in one of the SWI/SNF components, and prostate is one of the cancer that doesn't, but we see alterations. So I think you're absolutely right. It's a mechanism, just like the polycomb complex is also an epigenetic mechanism for dysregulation in a number of cancers. So I think these are examples where, you know, basic science, translational science, and clinical science have to all come together to really design and focus on perhaps the best ways to treat patients in the next setting.
Okay, brilliant. Thank you very much. So if anyone has any questions, just save them for the panel discussion.